Recording on the computer, share screen. Did my PowerPoint go? Did I not open it? Don't share on the PowerPoint. Oh, there it is. Didn't open it. Okay, back to Zoom. Recording, share screen. Now we got it. Okay, good. All right, so this is Unit 6. This is viruses. We're going to hit them again next semester in a more detailed way. We're going to hit them molecular and serology testing way this semester. So you'll get a double dose of virology. So we'll take it pretty slow, I think, through the first part, and then we have separate sections for an HIV on next week. So we're gonna cover viruses for this unit. I think this unit is all viruses, I think, up to the next test. And then we'll be doing testing downstairs for the antibodies and antigens that we can next week. So we're going to take a look at the virus structure and life cycle, the immune defenses. We'll spend a lot of time in that, immune defense against viruses, how viruses escape the immune system. Then we'll head into a little bit about laboratory testing. There's not a ton in this chapter on it, so we'll pick up individually. Laboratory testing, then we go into the hepatitis viruses. So we'll cover Hep A and E, BDC. Then we'll move into the herpes viruses, which are Epstein-Barr, cytomegalovirus, and varicella. And then we'll finish up with rubella, rubeola, mumps, and HTLV 1 and 2. So don't know if we'll get through all of it today. That's fine. I think I left myself some room in this unit because we're kind of running, if you hadn't noticed, we're kind of running out of chapters in the text. I think we're in 23, and those that like to look, we basically have HIV, immunization and vaccines is 25. Then I think we head back, can't remember. If we head back into some of the tumor, I think that's the last unit though. So I think we're going to stay pretty well in the viruses. So some good stuff. So let's take a look at the virus structure. If you hadn't seen pictures of a virus since March, I don't know where you've been. So you should see all kinds of goodies. They are submicroscopic, so we're not going to be able to take a body fluid and put them under the microscope and downstairs and look. So most every image that you ever see of a virus is electron microscopy. So those are kind of where those come from when you see them up close and personal. They either have a core of DNA or RNA. So we classify them that way. We'll say this is our DNA viruses, these are our RNA viruses. So as you see over here, that's inside this scientific looking structure, the icosahedral. They have protein coats, they have a capsid, and then they have an outer envelope, and that's usually where you see most of the goodies, like envelope proteins with coronaviruses. Those stick out like crowns, so that's how it gets its name. But that's a virus. So hopefully you've seen this before. So hopefully you understand that there is DNA or RNA in them and what that means. So we'll take a look at them. So I think, we start off by calling them obligate intracellular pathogens. So anything obligate is what? It's a must, right? It's a must. It must find a host. It's not going to survive. We've heard all about the timetables of is coronavirus on our feet? Is it on the floor? Is it on the keypad up here? Is it, where is it, right? So let's wipe it all down, and hopefully we won't have any transfer. You know, what they fail to tell you, I guess it's updates really, more so than anything else. Give me an update. Last update I heard was that coronavirus is unable to spread from fomites. Okay, that came from Fauci. So I kind of got to go with that. It was months ago, but we're still what? Wiping the heck down of everything that we touch, grab, everything. It needs your hands. Let's do it all. But what it's seen is it can be detected from the ground or your shoes or the doorknob, but it's not enough to transfer the illness. Okay, so that's kind of, as we say, fatigue is set in. That's where we are. We're not either focused on that, or we're not listening to that kind of stuff, or we're not willing to change what we're doing. We're just going to keep on. We bought all these supplies, so we're going to keep wiping everything down. But I think we need to think that, because we see the numbers. I mean, the number of cases have done that, and they're still up there. So until everybody gets exposed, we still have potential of getting it right now. So stay doing the things we know that work, which is, if you think you've got it, cover your mouth and stay away from people, right? Simple as that. So the virus structure and life cycle, they are obligate intracellular pathogens. So once they leave a cell, they try to get right back in. So our immune system is going to play on that. Our immune system is ready for that. And that's how the immune system will react first, is prevent that from happening. The virus, on the other hand, is going to do right opposite. The virus is going to figure out a way to get back into the cell, what it can do once it gets there, to hide from the immune system. So it's a cat and mouse game with viruses, which you're well aware of. So the virus has to rely on a host cell for replication and survival. And just think of that. I know I've mentioned this, and it's fascinating to the sound of that. These things, if you want to call them that, things, are they living organisms? Are they classified as living? If they can't survive on their own, right? So start there, and your little tweak, little wheels turning, all right? And then just think about what they do. Think about the mechanism they do. They have the ability to invade our cells, just basically reprogram our cells for their benefit. Okay, so to me, if you've ever watched any movies, I always like to say that they are evil, they're alien. I don't know where they came from, okay? But they're here, and they have a way of survival that's unmatched. Okay, so we're going to go through that process. We'll talk a little bit about it. But as you see, there's attachment. So they have a way, that external what? These envelope proteins, right? These envelope proteins link to the host cell's membrane of linking, right? They have that receptor, and they link, and then they're taken in. They invade the cell. They take part of the membrane with them, you see here, and then like they know they're in, so then they start undressing themselves, right? Just like alien movies to me. Just like, oh, I'm in now. Now I'm going to undress myself and invade your system. And they invade the genetic mechanism of the cell. just amazing that they have that ability to go in and say, okay, I know you, right? So here's a little bit of me, and this is going to help you produce things I need. So it's like mind control, take over the cell, invade the cell, the invasion of the body snatchers kind of idea here, right? And then they use that to start a new one. Then they reassemble, and out they go looking for another cell to invade. So that's the life cycle. And hopefully you've heard this before, kind of know how this has worked before. So I always like to put it into a correlation or example of car production. Like, we're making cars at this plant. Like, say I'm making the Tesla, kind of the newest thing, right? Tesla plant, I'm making Tesla. So I've got all my computers are designed to build parts, bring parts to the assembly line, and we put those parts together and out comes a Tesla. And then let's just say for fun, the Chevrolet company decides to invade the Tesla plant. Okay, the only way it can survive is to invade the Tesla plant. It invades the Tesla plant, uses the Tesla technology to build Chevrolet parts at the Tesla plant to assemble a Chevrolet that comes out the other end. Okay, it's not the Tesla at the other end anymore, it's the Chevrolet out the other end. Does that make sense? Hopefully you see it that way. So how complicated would that be? How smart would you have to be to do that, right? And these are viruses, and there are all kinds of different ones. Anyway, okay, so I hope I've got your focusing thinking. All right, so the immune defense, that's what we got. We got our immune system. It works better for some than others. Okay, so we have our innate defenses, is the skin and mucous membranes. So in that system, your textbook talks about, well, we have, if they're able to invade these barriers, okay, if they can get through the skin and the mucous membrane, the innate immune system recognizes by PAMPs, right, pathogen-associated molecular patterns on the surface of or within the virus-infected cell. So if they've invaded the skin, and we think about, just think about respiratory viruses, right? It doesn't take much. You get a big breath, inhale, virus is up your nose, and it's on the mucous membrane. You're hoping your defense will hold it there, but if it doesn't and it makes its way through, we have PAMPs. And then we have things known as interferon alpha and beta. So we talked about interferon weeks ago, and I told you one thing to remember about interferons is what? They mess with viruses. Okay, so their role is to mess with viruses. I think that's a simple way to put it. So two non-specific defense mechanisms against viruses involve interferons and natural killer cells. Okay. Virus-infected cells are stimulated to produce interferon alpha and beta following recognition of the viral RNA by the toll-like receptors. So the cell that gets invaded starts producing interferon alpha and beta. What's that do? What do y'all think? Interferons act how? They inhibit viral replication. How do they do that? They induce transcription of several genes that code for proteins with antiviral activity. And the example we like to talk about is ribonuclease enzyme that degrades viral RNA. So simply think about the coronavirus, okay? I know we're probably saving a special day for that, right? We're going to introduce all these others, and then we're going to kind of wrap up with corona. But it's an RNA virus, right? So when coronavirus invaded my cells flipped on the switch, produced interferon. That interferon maybe what kept me from showing signs and symptoms, maybe it kept me from being contagious, maybe it kept all that in check just by the innate immune system I had. Just the fact that it switched on and kept it from replicating. Okay, so it degraded the viral RNA. Alpha and beta also enhance the activity of the natural killer cells. And you know me, I love natural killer cells, which bind to the virus-infected cells and release cytotoxic proteins such as perforin and granzymes, and causing cells to die and to release that virus. Okay, before that cell has a chance to do what? Be taken over, to replicate the virus particles in its genome, to put out a new virus, to spit out a new one. Okay? So these free cell virions, now that the cell has destroyed, then the free cell virions are now out and about, and the rest of my immune system hammers it, right? So that's what we're thinking about. So now they're accessible to antibodies. Humoral antibody responses. So how many of you feel like you have been exposed to coronavirus previous to this new one? Who wants to raise their hand? Yay. All right, got one. Yes, more than likely you've had a cold caused by
coronavirus.
So is there any correlation? Don't have that detail yet, do we? We don't know. What if I went to the Red Cross and had my antibodies checked before I got infected? Did I have an antibody already? Maybe I have a similar antibody. Maybe I have at least some kind of recognition of the virus before this virus. Part of it, a finger of it, a toe of it, whatever way you want to think of it. Was there some kind of sensitivity already built up with people that aren't being overwhelmed by it, okay, or made sick by it, or running temperature by it, or coughing by it? I hadn't heard a cough yet. Have you? I mean, every cough I hear is negative tested, right? We had Dr. Walls coughing her head off for a week, went and got tested, negative. We had somebody else coughing, went and got tested, negative. So we don't, right? So it's very, very frustrating. Anyway, we're gonna make it. All right, humoral antibody response. The antibodies attack free virus particles that were exposed, right, by the interferons, the interferon alpha and beta, and the breaking up the cell, destroying the cell that was hosting that virus. So those antibodies, when they attach to those particles, they neutralize them. Of course, we've been talking about antimicrobials, right? Kind of in that same vein as the antibodies here being neutralizers. Opsonization is increased, so anything that has got an antibody attached to it, a phagocytic cell is going to like it more. Complement fixation, we got C3b, right, being plastered on top of these particles. And then we have our ADCCs, which are what? What are they again?
Antibody-dependent cell-mediated
cytotoxicities. Yes. All right. So that's all in play. So we know that we have IgG, we have IgM. They're there, right? IgG is there if it's been exposed to it previously. IgM is revved up on initial exposure. And then after all of that, we still have the next step, which is cell-mediated immunity comes in. And we bring in another interferon gamma, and interleukin 2. And these are produced by the T helper 1 cell. And the cytotoxic T lymphocytes, that's what CTL is there for, destroy the host cell. That's harboring the virus. It's holding on to the virus. Amazing, right? So this thing, that's come in, it's not like your immune system didn't have a chance. It was new, but it's not the first virus that your body's seen, okay? It was just a new one. All right, so here is a wrap up of that, everything that we just went through in a picture. Love the pictures, right? So you have your epithelial cell, that's your mucous membrane. Say that's your lung epithelial, is your skin, it's your nasal epithelial. And that virus has popped in. It's made its way through the first barrier. I see a ADCC. I have C natural killers. I see cytotoxic T cell. Okay. And these are the cells that the virus has invaded. That's trying to get it to replicate itself. Okay. So we have MHC. We got antibody. We have B cells over here. Who's this? That's a big plasma cell, right? Producing free antibodies. This looks like an epitopic cell dying. Apoptosis, right? Dying cell here, releasing viral particles for the immune system to either attach to, increase. So I think you got the idea. Hopefully you can make all of that out. If you can't, you can read figure 23.3. Kind of sums it up. All right, so now what happens to the virus? How can the virus, what makes a virus a good virus? What makes a virus a killer? Okay, so a virus can escape all those mechanisms that we just elaborately laid out. Everything that we just had learned from lecture 1 up to now, which is 26. Now the virus has a chance to show what it can do. And the first thing you want to know is it can mutate. Okay, since they replicate so fast, right? I'm in a cell, I unwind, I invade, you start producing things for me. So mutations, genetic mutations, since they're rapidly dividing, frequent gene mutations can occur. So the example, for those that have always wondered why in the world we need a new influenza virus vaccine every season, it's for this. Because the new, right, mutation results in a production of new viral antigens. So the vaccine that we got last year, now that antibody that was produced, or whether you got the flu from last year and that antibody that was produced for memory of that virus to come back, it's different this time. But it's mutated, okay? So you need some help, right? So what the help is the vaccine that gets developed each year is in response to the mutations that are seen from the previous year. So it says continual antigenic variation in the influenza virus results in the emergence of a novel infection strain every year. They require developing a new vaccine every year to protect the population. So that's what some people fear about coronavirus. They said, oh gosh, it's going to mutate. It's going to come back again next year as a little different one. And then it's going to come back again the next year as a little different one. Let's keep our fingers crossed, right? So, is the vaccine that's being developed going to be, by the time it shows up on the market, is it still going to be effective, or has Corona got a chance to mutate? So that's all on the table for the vaccine makers to understand. Do we have strain A, strain B? Do we have C? And is our vaccine going to work in different regions? Is it going to work if you got it on the West Coast, or you got it on the East Coast. And guess what? Just saw an update the other day. It says New York had it way ahead of when everybody thought they did. That's why it blew up in New York first. It was there a lot sooner than people realized. That's what they're saying. That's what they're saying now. So I think that's the story we're going to hopefully get updates on as we go through this. Everybody understand that? Why we need to get that new flu vaccine every year? Everybody got theirs already? Yay. All right, good. So you need to be, we also have rhinoviruses. That's another cold virus. And HIV, which we already knew. We knew that about HIV because it's been around now for what, 50 years? Not 50 years, 30, I was 18, 30 years. 1982-ish, 3-ish to now. So yeah, that's 30 plus years. And that's the biggest thing with it. It has mutated since the first virus showed up. All right. Any questions on that? That's important because that's key to understanding what's going on with viruses, the mutations they do. All right, here comes now that we mutate, right, and just give you a different antigenic structure or different antigenic look that you weren't really prepared for. Now, viruses can also block the action of the immune system components. How do this do that? An example they give is hepatitis C virus binding to C3b. Y'all know C3b is pretty important, right? Part of complement. So the second, the virus escape action components, complement proteins, right? For example, hepatitis C virus can block interferon-mediated degradation of the viral RNA, okay? So that interferon that we like, oh yeah, get after hepatitis C. Hepatitis C blocks it. Learned. Has it? I mean, that's what makes these like, how in the world do you have the ability to block something, right? Like, oh yeah, hey, that's important. And these are so little, right? This is not like a huge IBM big computer that's working out this process. Okay, so hepatitis C can block interferon. Okay, herpes simplex virus produce protein that binds to complement component C3b. Oh, that's herpes simplex virus, not hepatitis C. Sorry, HCV. I went hepatitis C virus, but that's herpes simplex virus for C3b. The hepatitis C blocks the interferon action. Herpes simplex virus, that's a mistake, isn't it? Yeah, that should be S on your PowerPoint. Sorry, I didn't catch that. That should be HSV, according to the text. Okay, so we'll trust the text and go with that. Anyway, we're talking about it between herpes simplex and hepatitis C virus. Blocking interferon sounds a lot like hepatitis C, and then simplex blocking complement 3b protein. You still good? Zoom, you still good? Let me ask it this way. Are y'all learning anything today? Yes? Or is this all new? Y'all know all this already? Or are you learning? Okay, good. Feel like it's worth it if you're learning. Suppression of the immune response, cytomegalovirus, reducing the major histocompatibility 1 expression. Read that, cytomegalovirus. Evade the host defense by suppressing the adaptive immune. Some viruses, such as cytomegalovirus, and HIV do this by reducing the expression of class 1 MHC molecules on the surface of the infected cell, making them less likely to be recognized by the cytotoxic T lymphocyte. Okay, makes sense, right? HIV goes dormant, hides out. We don't know where it went, right? It's not showing itself. The immune system kind of lays back and goes, I saw something flash by a couple of weeks ago, and now it's gone. But we do have the antibody that we can test for, and we'll talk more about this on Monday or Wednesday. I can't remember where we're going to end up with this. The cytomegaloid does the same thing, okay? Reducing the expression of the MHC1. So if you go back to that very first picture, right? If you don't have that expression, then you don't have the cytotoxic T cell's ability to recognize that it's invaded that cell. Other viruses, such as rubeola, can cause a decreased expression of class 2, resulting in reduced T helper cell activity. Keep that in mind, because we're going to hit rubeola eventually. It's in the back of this chapter. And then some viruses can alter the function of certain cells of the immune system by directly infecting them. Uh-oh. That's something new. Epstein-Barr virus, EBV. All right, immune function altered. So this is that number 4 up on the slide. Epstein-Barr virus. Well, before it could do that, causes polyclonal activation of the B lymphocyte. So what does that do? How's that change the immune function? What do you think? Epstein-Barr is over here, going over to 420. Just kind of get a preview before I won't get over there today. So go about halfway down, first column, a little farther than halfway. It says, Epstein-Barr virus enters B cells by binding surface CD21, which is the receptor for the C3d complement. The virus-infected B cells become polyclonally activated, proliferating and secreting a number of antibodies. Okay, that's where we are on this thing right here, stimulating polyclonal B cell activation, including Epstein-Barr virus specific antibodies, heterophil antibodies, and autoantibodies such as cold agglutinins, rheumatoid factor, and anti-nuclear antibodies. So it just, Epstein-Barr virus gets in there, the B cell just starts spitting everything out. In healthy individuals, the process is kept in check by the immune response of the natural killers and specific cytotoxic T cells. However, Epstein-Barr can persist in the body indefinitely in a small percentage of memory B cells, where this establishes a latent infection. Does anybody want to associate that latent infection to anything? Because this is where we get the narrative of talking about Epstein-Barr with something else. It's been dismissed for a long time. Now you're the immunologist. You tell me. It's a latent infection, persists for a long time. What's associated with Epstein-Barr virus? This is mono. Okay. So what's associated with it? It continues on for a long time. Maybe you are just too young. Maybe your parents have talked about, I might be disabled because of this. Maybe your grandparents. Zoom, you have an answer? We'll revisit it again. Just seeing where you are with this B cell. Chronic fatigue syndrome. You ever heard of that? That's good. It's good. No, it's not good. No, chronic fatigue syndrome is the idea that once you have mono, that it really, really never goes away, that you continually feel tired and fatigued for long periods after the infection. A lot of people have written that off and said there's no proof that there is such a syndrome called chronic fatigue. Others will disagree with you. So it is up for debate still in my mind, in my book. So this idea of the immune system latent infection and continuing on producing antibodies to a level that we get what? We know anti-nuclear antibodies, right? Produced with autoimmunity. Is chronic fatigue part of an autoimmune result of Epstein-Barr virus infection? Keep that in mind. All right. The last one we have here is a latent state is established. And this is, we have this really, with varicella zoster. But we do have it with cytomegalovirus, we have it with HIV, this latent stage of once we have an infection, the virus goes and hides for a while. And then once something, some other result, some other infection, it says, it reactivates. And varicella zoster reactivates into what? Second time around, it's called shingles. Exactly. The reason shingles are such a painful area and give the patient a lot of issues is because that latent hiding spot is in nerve cells. Okay. So down a nerve ganglion. So usually the back area underneath the shoulder blade down to the side is usually a very hot spot to see when it comes back. And it comes back in a pattern down that nerve ganglion. So if that's where it hid when you had chickenpox, that's where it reemerges. All right, so we're going to finish up a little bit talking about, well, I don't say we finished. I think we can move. We got a few minutes. We got how much time? What do we got? It's 9:40. We got what? 25 more minutes. Let's go. Right? 25 more minutes to go. Doesn't this go an hour and 15? This class?
No, that's Tuesday.
Oh, that's Tuesday, Thursday. Okay, sorry. Laboratory testing for viral infections, and this is what we're going to get to do, right? A little bit downstairs. So we got some kits that we're going to use on this. We know we've done one, right? Which one have you already done? See if you remember doing it. Because I couldn't remember you'd already done it. I was looking for the other kit and found another kit. And I was going to bring it back out and test you again. But we can still do that. It's fun. What viral test have you already tested for? Tested each other for? The influenza. Yeah, that's a virus, right? Okay, it's not on our list in this chapter, but we have. I used to get a kit each year for HIV. I used to do HIV 1 and 2s. Very similar, very easy to do that. But what we do mainly, we can do serological testing, which we are well aware of, distinguish between current and past infections. So once again, if you hadn't already gotten this down, this is another reminder as to the pattern of infection based on the immunoglobulin M and G pattern. So IgM positive with an IgG positive, Current infection would be where I went with that. If I was IgG-M and IgG negative, definitely a current infection. And then what they give you the out with is saying if IgG is present, that may indicate a recent infection, right? That you've gotten kind of getting over it, but you definitely have it. What is the congenital infection? What is that pattern? Why is that in here? I thought it was important when I read it. Oh, that's a good point. I like it, just to say the least. I like the point because we really hadn't focused on that point, which is if an infant comes out and tests with IgM, okay, there won't be any IgG from the baby. It'll be mom's IgG. IgG crosses the placenta. But if a baby tests for IgM, that means baby was infected in utero. Okay, baby can produce IgM. So if you've got a baby that comes out with IgM positive or something, you're tested like cytomegalovirus because of some kind of look of defect, that mom got it during pregnancy, okay, then IgM is there. IgG will be mom's. But if a baby tests for IgM, you got to think congenital in utero infection while we were pregnant, okay? So I thought that was a great point of testing infants because what we usually say is what? No need to test infants because infants hadn't developed their antibodies yet, right? We hear that all the time. Oh, they need 6 months, right, to develop their own IgG. Yes, but not if they got infected early. Okay, so keep that in mind. I thought that was a great point to bring up the testing. If there's no IgM and there's only IgG, that tells us that's a past infection, right? That tells us IgG's there, means you had it, you produced an antibody, but there's no IgM level detected. That means that it's old. Not, you're way away from the initial infection because IgM was the first and died out, went away, but IgG stayed for memory. We can do antibody titers used to monitor course of infection. This would be important when I had a coworker that got Hep C during work, and one of the things that we did to monitor her was run antibody levels. We also ran viral loads, okay? So we could do both. Also, we drew her blood, we checked viral loads, we checked antibody loads. So doctor would monitor that, seeing how she was doing, because it's chronic infection, chronic liver, hepatitis, and we'll talk more about that as we get to C. We can also assess our immune status. Virus-specific IgG indicates immunity. Yay, right? That's a fun time to pull out your phone and show people, right? That's what I've been doing. Right? Wouldn't it be fun if we did that for everything? Like, what do I have immunity to? Do I need to worry? Do I have immunity already, right? What's scary, though, is when people go get that test and it's not positive yet for COVID antibody, because they're still susceptible, okay? Or more susceptible than one that would have antibody. So yes, we can assess immune status. We also do that with Hep B, right? You went and got your Hep B vaccine, or if you didn't have it when you were little, you had to go get the 3 shots again before you go to clinical. And part of that assessment, just so you know, I went through the assessment because I got it in school and college and it didn't work. I didn't produce an antibody to Hep B at the time. We had to do it again. So I had to go back through the Hep B vaccine protocol one more time. Then my level was positive. We did our levels again and it was positive. So we can do that. So when you get that option, you can say, well, you need to take the vaccine, or if you think you've already had it or you've already been exposed, then go get an antibody level and it'll tell you whether you have a level high enough to infer what? Immunity. So that's always good. And then to finish out, we have molecular tests. And hopefully you've heard a lot about this, right? So molecular testing detects an active infection. Quantitative tests guide antiviral therapy. But molecular testing, your book has a good little section on And this is where it confused me. Remember I told you I was confused over the RT-PCR? Well, your book confuses me again because it starts off calling it reverse transcriptase PCR, which is RT-PCR, right? And then it turns into real time. It goes from reverse transcriptase to multiplex real-time PCR, RT-PCR. Aren't those the same initials? Am I missing something? Is RT-PCR and RT-PCR all the same? Anyway, so it is confusing when you see those two. It gives you a little connection. It says in reverse transcriptase PCR, viral RNA is treated with enzyme reverse transcriptase to generate a complementary DNA, so cDNA sequence. The cDNA is then amplified by polymerase chain reaction to generate millions of copies that can be detected in the lab. And we did that in chapter 12. Remember PCR testing. And that is our big confirmatory test that we're doing still today is PCR. So coronavirus is our RNA. So they denature the virus. They take a sample, take your swab. They denature it. We had the ability, we're doing it over in ABI. They have the readers and the test kits over there. It's kind of been replaced by the antigen test since it's quicker. Antigen test takes 15 minutes, where reverse transcriptase PCR takes about an hour. Okay, so you can see if you had 1,000 people test today, you got 1,000 swabs to go through. Real-time PCR is going to take a while. Okay. So that's the description. I like that little short description it just gave. I know we're running out of time for today. I think we did well. So we'll crank up with the hepatitis viruses when we come in on Monday. I think you have the important part from an immunologist point of view. And now the rest of it's just learning or reviewing. I won't say learning. I'm going to say reviewing these infections that you've already heard about many, many times. All right. Y'all have a good weekend. We'll stop the share. We'll say bye to the Zoom crowd.